And we have Victor Perez up next.
Hello, everyone. First of all, thank you to the organizers of the summit and the hackathon. It's my first time in the summit, and it's been a very enjoyable experience. So I want to talk to you about an image analysis pipeline that is used in multiplexed imaging and spatial omics. But before going into the topic, I would like to thank my colleagues and collaborators in this project. So my colleagues from the Institute for Computational Biomedicine in Heidelberg, external collaborators in Tübingen, in Sequera Labs, and as well to the original development team of MCMICRO. So as I said, I want to talk to you about an image processing and analysis pipeline, but I also want to show you the context in which this pipeline lives. So I will make an overview of what multiplex imaging and spatial omics is. Then I will guide you through the pipeline step by step. And finally, I will show you the work that has been done to transfer this pipeline into NF-Core. Okay, so what we're seeing here is a small region of interest in tissue. The first goal of spatial omics is to study cell interactions, not in small regions of interest, but rather in whole tissue. Yeah? So the second aim is to investigate the interactions in space of different cell types. And for that, we need to phenotype these cells. That means we need multiple markers to assign the cell types. And this is done through the technology called multiplexed imaging. So in multiplexed imaging, or multiplex imaging is a family of methods that allow the cyclic staining of samples. One can have multiple cycles in a single run, and within each cycle have 3 or 4 markers. So at the end, one ends up with 20, 40, or even hundreds of markers in the same sample. Just as a personal anecdote, I was in the field of microscopy 5 years ago, and back then people was happy with 4 or 6 marker stops, yeah? So the progress of multiplex imaging, it's just tremendous, yeah? So the data set that I showed before, it's a 20-plexed image. That means it's composed by 20 channels or markers. And now I'm going to show you what is the general framework of spatial omics. So we take our multiplexed image, we use the channels staining the nuclei and the membrane to perform segmentation of cells. Segmentation means locating the cell and also getting to know the area that occupies in the tissue. Once we have isolated the cells, we use the expression patterns of the markers across all channels to assign phenotypes to these cells, yeah? Okay, so this is more or less the framework of spatial omics, but I also want to show you what is the relevance of the field. I will change the sample, just hold on there. So I'm showing you now tumors in breast tissue, yeah? There are 3 tumors here. They're classified as mixed, compartmentalized, and cold, yeah? There are 2 types of cells there, immune in orange and tumor cells in blue. So the classification has been done using the information acquired from the spatial organization. For example, the mixed, if we only look at composition, the mixed and compartmentalized tumors look basically the same, yeah? So there is no extra information there. But thanks to the spatial organization, we can see how the cell types interact and create new classifications of these tumors. So the relevance of this is that it opens the doors to patient stratification and also potential custom treatments. Okay, so this is the framework where the pipeline exists. Now, what do we need to implement it? So we have a bunch of algorithms, and each of these algorithms have its own required libraries and packages, yeah? We also have to deal with the different output formats, yeah, from all the new technologies that are coming out there. So even if we have all these variables, there is a blueprint for this pipeline. And I'm going to go through the pipeline step by step. Yeah? So let's imagine that one of these devices produces tiles as raw data. As I said, we are interested in investigating tissue, whole tissue images. So we need to stitch these tiles to have a single image. Now, as you might see, the image gets dimmer in the edges of the tiles, and this is due to the inhomogeneity of the illumination. This is just a limitation of the microscope, so we need to correct for it. Yeah. And once we have applied the illumination correction, we go to the step called channel registration. So we need to remember that we have different channels in a data set. These channels are acquired in different cycles, so it's very likely that the sample will drift during acquisition. Yeah? So we need to correct for this drift and align all these images in the XY plane. Once we have the channel registration, we can also enhance the contrast of the image by correcting the autofluorescence. Yeah. And finally, once with the enhanced contrast, we can apply segmentation algorithms. For that, we require the nuclei channel. So we segment the cells. And finally, we can extract quantifiable features that will help to do later the spatial analysis and the phenotyping. So this pipeline, the blueprint of this pipeline, has been already assembled in Nextflow. This is a paper from 2021. It's Dennis Shapiro, my supervisor. Yeah. But now we want to go to the next step of standardization. So we want to have our pipeline curated. We want to have it tested, and we also want guidelines to include new modules in the pipeline. So the natural step is to go into NF Core, and we've been working lately to do this. What we've been doing so far is first include the modules that are already marked with green. Yeah? These modules are already NF Core modules, and we are using them to assemble the whole pipeline. This is currently work in progress. We will be using the sample sheet to handle multiple data sets in a single run. We also want to have optional modules. That's why it's MCMICRO, multiple choice. Yeah. But also we want to take into account the devices that are, the acquisition devices that are out there. So we would like to include staging modules before injecting the data set into the pipeline. Yeah. So I would like to have this, my aim is to have this ready by December, yeah. The pipeline and at least one of the staging modules. And before ending my talk, I would like to do just some shameless promotion. My colleague Krešimir Bestek is presenting a poster in stand 9, look for him. It's also an image analysis pipeline. He's in the very corner. And if you're interested in image analysis pipelines, just reach out and let's connect. Yeah? Thank you.
Thank you very much, Satish. That was fascinating. We have a few questions out here. One is, what was the most challenging in bringing the non-genomics pipeline to NF Core?
In bringing what, sorry?
The non-genomics pipeline to NF Core. What do you think were the biggest challenges in that?
I think it's finding the right algorithms for it, yeah? Because you could add as many modules as you want, but yeah, we want to keep this blueprint simple, yeah? So not to have so many tunable parameters. Yeah. So it's conceptual. The challenge, it's more conceptual, yeah?
The other question is more specific. What is the limit on number of channels? How many colors can be distinguished?
Well, so far, I've worked even with 100 channels, yeah. I can just talk from experience. I've seen 20, 40 channel data sets, but also I've worked with 100 data sets, yeah, 100 channels data sets, yeah.
We have a question from Ben. Are there any potential Nextflow features that would make it easier to develop and run your image processing pipelines?
Any Nextflow features? Well, I don't know if, so as I said, the pipeline, it's already assembled in Nextflow, yeah? We were working with it for about a year, but we realized that we need guidelines for it, yeah? And so those tools are currently provided by NF Core more than Nextflow, yeah?
What is the most challenging step in the pipeline in terms of algorithmic complexity?
In terms of what, sorry?
Algorithmic complexity.
I mean, all these modules do a different thing, yeah. I won't be able to only mention one. Yeah. Each of them make their own task, and they do it well, yeah. But what is more complex, it's difficult to rate that.
Is it possible to run the same analysis manually on a researcher laptop? Or what are kind of benefits of performing this sort of headless image analysis with Nextflow?
Well, I mean, that will depend on the size of the data set, right? And the size of these data sets can be up to 1 terabyte, yeah? So it goes from 500 gigabytes to terabytes. Yeah? So I'm not sure if you can just simply perform that in your laptop, yeah.
And then, are there any of the tools used by the pipeline optimized for GPUs specifically?
No. We're working on implementing that. Yeah, we're working on implementing that because not all modules require GPU, so we will have to configure the process that really require GPU. Yeah.
Great. And maybe one last question: is there any other pipelines already available or packages to do the same?
Not really. No, as far as I know, there is.
So that's the first one.
Yeah.
Great. Thank you so much.
Thank you.